Hello everybody, it's Ariz and I'm back with another video. Today we are going to be focusing on the second chapter of GCSE's biology, that is diffusion, osmosis and the concept of surface area to volume ratio. So let's start. So first up we have diffusion. Diffusion is the net movement of molecules from the region in which they are in higher concentration to the region in which they are in lower concentration down the concentration gradient. This over here is a concentration gradient. You see there is net movement of molecules down the concentration gradient. They usually ask in paper 6 to design an investigation to demonstrate diffusion. So this can be one. Over here I've taken copper to sulfate crystal and dropped it in water and observed the blue color spreading. And you see there would be blue color spreading from the region in which it was in higher concentration, that was at the lower part, to the region in which there was in lower concentration, that was the upper part. Next up we have osmosis. Osmosis is net movement of water molecules that takes place from the higher concentration to lower concentration, and remember guys, there is always a partially permeable membrane in between. Osmosis also takes place down the concentration gradient. To demonstrate osmosis, you can design an investigation like this. You have 10% sucrose solution at A side and 5% sucrose solution at B side. Notice the water levels are the same, and after a while you would notice that there was a rise in water level at A because there was net movement of water molecules against down the concentration gradient through a partially permeable membrane. There is another important concept of hypotonic, isotonic and hypertonic solutions. Hypotonic means higher water potential, hypertonic means lower water potential, and isotonic means equal water potential as compared to the water potential of the solution we are comparing it with. Now this table over here guys is extremely important. Let's discuss it. Over here we have a plant cell. When we put it in a solution that has a higher water potential, what would happen? The water would move from the region it was in higher concentration, that was outside, to the region it was in lower concentration, that is inside the plant. As this happens, plant cells swell. This is because water has moved inside. And notice that it does not burst because we have cell wall over here that protects it from bursting. Similarly, if we put an animal cell in a solution having higher water potential than the animal cell, then water would move inside by endoosmosis and the animal cell would burst because it has no cell wall. This situation of plant cell is described as turgidity, whereas the situation of an animal cell is simple bursting. When the plant cell is placed in a solution containing lower water potential, water would move out of the cell by the process of exoosmosis. And you have to notice some things over here. For example, the cell membrane becomes detached from cell wall. This is because cytoplasm and the cell both lose water. Water is moved out. So the cell membrane is detached, and they mostly ask in paper 1 to label the cell membrane. You won't be labeling this, you would be labeling this. And when an animal cell is placed in a solution containing lower water potential than the animal cell, it would lose water as well, but it will crinkle, and the situation is known as crenation. This was one of the most important concepts. I hope you have understood it right. Let's discuss the importance of turgor in a plant. Well, first of all, it maintains the shape of soft tissues in a plant, for example, leaf. Because of this, the non-woody plants mostly can be upright and gain maximum sunlight. The build of turgor pressure controls many plant processes. For example, the process of wilting when there is excessive loss of water or a decrease in turgor, or the curling of mimosa leaves when you touch them. There is another alternative process to diffusion and osmosis, that is active transport. Now, active transport is a process in which there is net movement of molecules from the region in which they are in lower concentration to the region in which they are in higher concentration against the concentration gradient. Okay, there is an investigation that repeatedly comes in the investigations of osmosis. So in here you are going to take a tape or a stalk. Over here we have the Chinese leek. You would cut it into 4 layers, and you would observe as soon as it is cut, it forms curvatures. Now the reason for this is the outer layer of a leaf is epidermis, and it has a layer of cuticle on it, the waxy substance, so there is no net movement of water molecules in and out of the leaf. Whereas the inner layer is cortex, it has no such thing as cuticle, so it can lose or gain water. So when this is cut, there would be no loss of water, but there would be gain or loss of water in this layer. So this causes a curvature to form. For example, in a bimetallic bar you have studied in physics, it happens because of their different expansions. One expands more than the other, and there is a curved form. This is because of this. Okay, the last concept we have to cover over here is the concept of surface area to volume ratio. Now see, over here this little box has the highest surface area to volume ratio because, for example, it has 4 sides on which it can react and produce its products. Over here, when the object becomes bigger, over here it only has 2 sides to perform its function, and over here it only has 1 side. So as the object becomes bigger, the area becomes bigger, the surface area to volume ratio decreases. That's why elongated things increase the surface area to volume ratio. Okay, that was all about the video. Hope you liked it. Bye.